My name is Nina Cristal from BSIE 1301 and I will be reporting about the cell cycle. So before we proceed on the actual presentation, let us first discuss the terms to remember when it comes in understanding the cell cycle. The first one is sister chromatids. It is the identical pairs of DNA molecules that are firmly attached to the centromeric region. And as you can see in the photo, it is binded by a centromere. And it is merely the result of the DNA replication during the S phase. Next is the mitotic spindle. It is the apparatus that orchestrates the movement of chromosomes during mitosis, and basically the product of the two centrosomes. The next one is the chiasmata, a point at which paired chromosomes remain in contact during the first metaphase of meiosis, and at which crossing over and exchange of genetic material occur between the strands. So, as you can see, it is the vertex of the two chromosomes after the process of exchanging genetic material or yung tinatawag nating crossover. The fourth one is the centrioles. It helps organize cell division. As you can see, it looks like a rod-like object which is associated in the centrosomes of the animal cell. And another fact, present din sila sa other eukaryotic cells such as plants and most of the fungi. The next one is the kinetochore, a protein structure that binds and attracts mitotic spindle microtubules, and they are basically developed by sister chromatids. The sixth one is the polar microtubules. So it is defined as the spindle microtubules that do not engage the chromosomes, and they do not engage. Instead, they overlap each other between the two poles, midway between the two poles, which would then contribute to the cell elongation. Next is the metaphase plate, what we also call the equatorial plane, where chromosomes are aligned. And as you can see in the photo, this is where a series of chromosomes are organized or perfectly aligned. The other thing is the cleavage furrow, which is the fissure or crack that deepens as the actin ring cracks. Typographical error lang po, pero it should be ring. And this happens in the process of cytokinesis. Before the cell actually divides, it doesn't divide like this, but instead, cleavage furrow will happen first. It will form a curve like this, then divide. And later on, it will be further discussed. Next is the cell plate. It is the structure in which vesicles fuse and coalesce from the center toward the cell walls. So, this enlarges as more vesicles fuse until it reaches the cell wall. So, palaki siya ng palaki, pa-expand siya ng pa-expand hanggang sa ma-reach niya yung cell wall. The ability to reproduce in kind is a basic characteristic of all living things. So, in order to reproduce by sexual reproduction, fertilization is required, and it is defined as the union of two cells from two individual organisms. So we've understood that in fertilization, there is a need of the union of two cells, and individually, each of the cell contain one set of chromosomes, and that is what we call haploid cells. While on the other hand, when combined, it will form the diploid cells, which contains two sets of chromosomes. The haploid cells and the diploid cells is what we call the ploidy level, or the number of sets of chromosomes in a cell. Next is the homologous chromosome. From the term itself, homologous means same structure, and it is defined as the matched pairs containing the same genes in identical locations along their length. And this is basically found or nakikita sila sa nucleus. Next one is the spores. They are the haploid cells that can produce a haploid organism or can fuse with another spore to form a diploid cell. So, it can produce some plants and all fungi. And makakaproduce lang siya ng diploid cell kapag it fuse with another spore. Parang spore plus spore, saka palang makakaproduce ng diploid cell. So next is the cohesin. They are the proteins that held sister chromatids together at the centromere. So it holds the chromatids until the anaphase 2. So for now, siguro hindi nyo pa nagigets or familiar na kayo sa mga sinasabi kong mga phase, anaphase, S-phase. Later on, it would be discussed by my other group mates para sinasabi ko lang itong mga terms na to para pagka nandun na kayo sa pag-understand kung ano yung cell cycle, alam niyo na yung mga terms para mas madali niyo siyang maintindihan. Next is the synaptonymal complex. It is a lattice of proteins between the homologous chromosomes. So at first, it forms in a specific location or sa isang certain location, and then it spreads. So it would cover up the entire length of the chromosome. So let's proceed to synapsis. It is a tight pairing of homologous chromosomes, and in synapsis, the genes of the homologous chromosomes are aligned precisely with each other, as you can see on the photo. Next is the recombination nodules. It is the large protein assemblies located at intervals along the synaptonymal complex. So these mark the points of later chiasmata, which would result to the next, which is the crossover, the genetic recombination between the non-sister chromatids. And lastly, the kinetochore microtubule, a spindle fiber that has attached to a kinetochore. That's all, thank you, and I hope these terms would help you in understanding the cell cycle better.
Good day, I am Erwin Bernard Talento II, and as you can see from the presentation, we are from Group 2 going to discuss the cell cycle. So what is cell cycle? So cell cycle is an orderly sequence of events that describes the stages of a cell's life from the division of a single parent cell to the production of two new daughter cells. In short, cell cycle allows the growth and repair of cells and also the division of cells. So as you can see, the cell cycle has two major phases, which are the interphase and the mitotic phase. So later on, these two major phases will be further discussed by my group mates, but for now, I am going to give you a brief overview of what these phases are. So the first one is the interphase. So during interphase, the cell grows and the DNA is replicated. In this phase, you will be able to encounter familiar terms like G1, G2, G0, or S for synthesis. And during mitotic phase, the replicated DNA and the cytoplasmic contents are separated and the cell divides. So in this phase naman, you are going to encounter terms like prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So let us move on to the interphase.
Hi guys, I'm Christian Ranz Balmas and I'm here to present to you what is all about the interface. If you hear the word interface, what comes in our mind first? So, interface, let's recall about the meaning or some knowledge regarding the interface. So, interface is the starting stage of the cell. Interface has three stages, and that are the first gap or the G1, the second gap or the G2, and the S phase or what we call the synthetic DNA or the synthetic of DNA. And as far as I know, interface is the longest stage in the eukaryotic cell. Dito sa interface, ito yung part na kung saan nag-grow yung mga cell. Dito din nagre-replicate yung mga DNA at nagre-repair ng cell. So, ano nga ba talaga or what is the real meaning of the word interface? It is the cell undergoes normal growth process while also preparing for cell division. So, ito yung pinakamahaba na cycle that occurs in the eukaryotic cell. What is the meaning of eukaryotic cell? A cell that has a nucleus. So, ito yung cell ng mga living organism. So, yun yung eukaryotic cell. Then, interface has its function. And what is it? And what is its function? During interface, the cell copies its DNA in preparation of mitosis. So, sa interface pala, nagkakopy yung mga DNA. So, I will proceed the function of the interphase. Interphase is the daily living of metabolic phase of the cell, in which the cell obtains nutrients and metabolizes them, grows, reads its DNA, yun nga, din pala binabasa yung mga DNA, and conducts other normal cell function. So, the three stages of interphase is the first, or the G1 phase, or the first gap, the G2 phase, the second gap, and the last one is the S phase, or what we call the synthesis of DNA rather. So, what is the G1 phase, or the first gap? So, phases of the cell that takes place in the eukaryotic cell division. So, that is the G1 phase. And the second phase is the G2 phase, or the second gap. Dito naman, it is the final step of interphase. Then, during this step, the cell makes everything still needs to divide. So, dito naman pala, ang lahat ng cell ay nangangailangan or nanatili na magkakahiwalay sa second gap. The last one is the S phase, or the synthesis of DNA. In this phase of the cell cycle, in which DNA replicated. So, dito nakakopya occurring between G1 phase, or the first gap, and the second gap. So, after this, my group mate will discuss all about the mitosis, the meiosis, and other parts of the cell cycle. So, I hope you learned something regarding the mate report, which is the cell cycle. And stay safe, everyone. Then, God bless.
Good day everyone, this is R.S. Astrillo. I'm going to present about mitosis. Mitosis is a type of cell division that results into two daughter cells, each having the same number and kind of chromosome as the parent nucleus and typical of ordinary tissue growth. So let's go to mitotic phase. So nakalagay dito, mitotic phase is a multi-step process during which the duplicated chromosomes are aligned, separated, and moved to opposite poles of the cell, and then are divided into two no identical daughter cells. So, mitotic phase, guys, has two portions. The first one is the karyokinesis, or nuclear division, and the second one is the cytokinesis. Let's start with karyokinesis. Karyokinesis is divided into 5 phases, which are the prophase, prometaphase, metaphase, anaphase, and the last one is the telophase. So let's start with prophase. So this is the illustration. As you can see, chromosomes condense and become visible. Spindle fibers emerge from the centrosomes, nuclear envelope breaks down, and nucleolus starts to disappear. Okay, so next one, the prometaphase. This is the continuation of prophase, which is the chromosomes continue to condense, kinetochores appear at the centromeres, mitotic spindle microtubules attached to kinetochores, centrosomes move towards or starting to move towards the opposite poles. Okay, so let's continue with metaphase or change phase. So, bakit siya tinawag na change phase? Kasi, kita naman natin yung pinagkaiba ng illustration niya, yung ngayon, kaysa dun sa kanina. So, mas visible na yung differences nila. So, actually, metaphase is particularly used in cytogenetics. Bakit? Kasi, it is the most easily visualized stage of mitosis. And then, sabi nga dito, mitotic spindle is fully developed na. Centrosomes are at the opposite poles of the cell. Chromosomes are already lined up at the middle of the metaphase plate. Each sister chromatid is attached to a spindle fiber originating from the opposite poles. So, ayun. The next one is the anaphase. The progression of cells from metaphase into anaphase is marked by the abrupt separation of sister chromatids. So, the reason would be cohesion proteins binding the sister chromatids together break down, Sister chromatids are pulled toward opposite poles. Non-kinetochore spindle fiber lengthen elonginated the cell. So dito nagsistart na silang maghiwalay and then tinutulungan siya nung spindle fiber na bumalik or pumunta dun sa magkabilang pole. And then mitosis ends with telophase, or the stage at which the chromosome already reached the poles. So the nuclear membrane then reforms, and then the chromosomes begin to condense into their interphase conformation. So ito na yung last na part ng mitosis or last na stage. Let's move on to cytokinesis. Then telophase is followed by cytokinesis, or division of the cytoplasm into two daughter cells. The daughter cells that result from this process have identical genetic composition. So dito naman, we have two cells, the animal cell and the plant cell. Okay. In animal cells, a cleavage furrow separates the daughter cells. So, ganun yung process niya. Sa plant cells naman, a cell separates the daughter cells. That's all. Thank you.
Now that you are familiar with the interphase and mitosis, let us have an overview of what the whole cell cycle looks like. So as you can see in the picture, the three stages of the interphase are present, which are the G1, S, and G2, and also the M, which stands for mitosis. And as you can see also in the picture, there are checkpoints. So checkpoints in the part of the cell cycle. So checkpoints are what it literally means. So checkpoints checks the cell if it met that certain requirement. And I know this hasn't been mentioned earlier, but there's also another stage of the interphase that hasn't been mentioned earlier. So which is the G0 or resting state. So instead of the cells proceeding further in the cell cycle, some cells proceed to the G0 or resting states like the neurons or the nerve cells. So this also might explain why when our spinal cords get damaged, it does not repair. So it explains that the neurons or the nerve cells, instead of going through the cell cycle in order to repair the damaged cells, it rather proceeds to the G0 or the resting state, wherein they are just in the G0. So now let us move on to meiosis. So where does meiosis comes in? So meiosis is also similar to mitosis, but of course they have their differences. So to clarify out their differences, we have this picture. So meiosis and mitosis are involved in making new cells. So that's their similarity. But for mitosis, so a cell cycle with mitosis makes body cells, which are the stomach cell and skin cell, while cell cycle with meiosis makes gametes, or which we know as the sperm cell and the egg cell. So before meiosis, we also know that there is an interphase happening, so which replicates all its DNA and organelles in preparation for the division. So meiosis has two parts, which are the meiosis 1 and meiosis 2. So we will first discuss what is happening in meiosis 1. So in meiosis 1, there are also similar parts to the mitosis. So first, we have the prophase 1. So in the prophase 1, chromosomes condense and the nuclear envelope fragments. Homologous chromosomes bind firmly together along their length, forming tetrad. So chiasmata form between non-sister chromatids. Crossing over occurs at the chiasmata. Spindle fiber emerge from the centrosomes. So it's also similar to the prophase in mitosis. But to further understand it, let us proceed to the picture before prometaphase. So here, as you can see, the spindle fiber, so which are yung parang mga yellow wires which grow out of the centrosome. And as you can see, the nuclear envelope starts to dissolve at prophase 1. And as you can see, there are chromosomes. So the chromosomes also condenses here, like in mitosis, but the difference here is that now chromosomes here match up with their homologous pairs. So what are homologous pairs? So homologous pairs are those which has the same size and the same type of genes. So as they have paired up with each other, there's a chiasmata which connects them, and there's when it connects there is also a process called the crossing over which is happening, which the chromosomes transfers genetic information. Next is the prometaphase 1. So here in the prometaphase 1, homologous chromosomes are attached to the spindle microtubules at the fused kinetochore shared by the sister chromatids. Chromosomes continue to condense and the nuclear envelope completely disappears. So here in the prometaphase 1, yung mga yellow wires na nakita natin kanina, nagko-connect na dun sa mga homologous chromosomes. So, yung kinetochore is what yung parang nagko-connect sa chromosomes sa mga spindle fibers or spindle microtubules. So, as you can see in the picture before metaphase 1, yung mga yellow wires nagko-connect na dun sa mga chromosomes at their centromere. Metaphase 1, so homologous chromosomes randomly assemble at the metaphase plate where they have been maneuvered into place by the microtubules or the spindle as the spindle microtubules. So in the metaphase 1, so similar din to sa mitosis wherein nadadala sila sa gitna ng mga spindle microtubules. But in mitosis, so they are aligned in the metaphase plate or yung gitna ng cell into a single file. So dito naman sa meiosis, they are also arranged in a single file but they are arranged in pairs. So they are arranged in pairs in the metaphase plate. So as you can see in the picture before the anaphase, so the homologous chromosomes are arranged in the middle by the spindle microtubules. So they are arranged also in the single file, but they are in pairs. And look, pay attention to what is happening to the picture. So this is what's going to happen next in the anaphase. So anaphase, spindle microtubules pull the homologous chromosomes apart. The sister chromatids are still attached at the centromere. So yung mga yellow wires, pinupull away nila yung chromosomes pinupull nila yung homologous chromosomes sa magkabilang sides. So they are parang sineseparate na niya. And we will proceed to telophase 1 and cytokinesis. So sister chromatids arrive at the poles of the cells. So yun nga yung nahilangan ng sa anaphase, naka-arrive na sila sa magkabilang poles ng cells. So, wherein they begin to decondense. A nuclear envelope forms around each nucleus and the cytoplasm is divided by a cleavage furrow. The result is 2 haploid cells. So, each cell contains one duplicated copy of each homologous chromosome pair. So, here in the telophase 1 is similar also sa mitosis. So, na kung saan nagpo-form na yung nuclear envelope around the chromosomes, and which will be followed by cytokinesis wherein yung cleavage furrow, so babalik tayo sa picture. So yung cleavage furrow ay yung nagdi-divide sa cell. So from a single cell, magiging dalawa yung cell. So yun, yung cleavage furrow is yung nasa middle nitong dalawang cell. Now that we know about the first stage of meiosis, which is meiosis 1, we will proceed to the meiosis 2.
Good afternoon everyone, I am Samantha Baliwag and I will discuss about meiosis 2. So, the 2 cells produced in meiosis 1 go through the events of meiosis 2 in synchrony. During meiosis 2, the sister chromatids within the 2 daughter cells separate, forming 4 new haploid gametes. In the events here in meiosis 2 is similar to mitosis, except that the daughter cells produced will only have half of their parents' genetic material. So the first is the prophase 2. Here, sister chromatids condense, a new spindle begins to form and the nuclear envelope starts to fragment. So in this process, they're not gonna have homologous chromosomes and the crossing over does not happen, just like in meiosis 1 sa prophase 1. Homologous chromosomes ay yung paired chromosomes na galing sa parent during fertilization. The sister chromatids make up the homologous chromosomes. And the crossing over of chromosomes exchanges segments of non-sister chromatids in the homologous chromosomes that results in genetic variation. Next is the prometaphase 2. The nuclear envelope disappears and the spindle fibers engage the individual kinetochores on the sister chromatids. So dito, tuluyan nang nawala ang nuclear envelope, and the kinetochores are then attached to the centromere of each sister chromatid. Next is the metaphase 2. Here, sister chromatids line up at the metaphase plate. So in this phase, the sister chromatids align in the middle but not in pairs, just like in metaphase 1. The sister chromatids prepare themselves to move at the opposite poles. Next is the anaphase 2. Sister chromatids are pulled apart by the shortening of the kinetochore microtubules. Non-kinetochore microtubules lengthen the cells. So, here in anaphase 2, the sister chromatids begin to separate and move to the opposite poles. Sometimes, anaphase 1 and 2 may not correctly separate homologous chromosomes and their sister chromatids, respectively. The failure to separate homologous chromosomes is called non-disjunction. This may have adverse effects depending on the chromosome that is affected. And lastly is the telophase 2 and cytokinesis. In telophase 2 and cytokinesis, chromosomes arrives at the poles of the cell and decondense. Nuclear envelopes surround the 4 nuclei. Cleavage furrows divide the 2 cells into 4 haploid cells. So here, in telophase 2, the chromosomes uncoil and the nuclear envelope reappear. Cytokinesis finally splits the cells, producing 4 haploid cells. Keep in mind that the product of meiosis are 4 haploid daughter cells, whereas the product of mitosis are 2 diploid cells. So, to sum up, nangyari sa meiosis 2 yung one set of duplicated chromosomes na nag-line up sa metaphase plate na may divided kinetochores na naka-attach sa kinetochore fibers from the opposite poles. So, during anaphase 2, the kinetochores divide and their sister chromatids, now referred to as chromosomes, is pulled to one pole while the other sister chromatids is pulled to the other pole. So kung di dahil sa crossover, yung dalawang products ng individual meiosis 2 ay magiging identical gaya ng sa mitosis. They are different now because there has been at least one crossover per chromosomes. So, meiosis 2 is not a reduction division because although there are fewer copies of genome in the resulting cells, there is still one set of chromosomes as there was at the end of meiosis 1. That's all. Thank you.
In conclusion, we can refer mitosis to the process of replication, wherein it produces 2 identical cells in which we refer to as diploid cells. So each diploid cell contains 2 sets of chromosomes, one from each of their parent cell, and meaning each diploid cell contains 46 chromosomes. While as for the meiosis, we can refer it to as the division reduction, where it produces 4 unidentical gamete cells. So for males, we can refer it to as the sperm cells, and for females, we can refer it to as an egg cell. And those 4 unidentical gamete cells have 23 chromosomes each, or which we can refer to it as a haploid. So haploid contains half of the chromosomes of their original starting cell. And we all know that sperm cell has 23 chromosomes and an egg cell has 23 chromosomes also. And that if we would put them together, it would form a fertilized egg, which we can refer it also as a zygote. So a zygote which will then have a total of 46 chromosomes, which will then also develop into a new organism. So I hope that you learned something from our report, and that would be all. Thank you.